Hello everyone and welcome to my YouTube channel, Perfect Lab. In today's video, we will learn about the Urease test, a simple but important biochemical test used to identify bacteria that produce the enzyme urease. We will also cover the principle of the test, the procedure, result interpretation, and its significance in bacteria identification. So let's get started. Start our introduction by looking at what the Urease test is. So the Urease test is a biochemical test which is used to determine whether a bacterium produces the enzyme urease, and it is commonly used to differentiate members of the enterobacteriocyte and to identify other clinically important bacteria. Principles of the Urease test. The Urease test is based on the ability of certain bacteria to produce the enzyme urease to hydrolyze urea. So bacteria that produce the enzyme urease hydrolyzes or break down urea into ammonia and carbon dioxide. The ammonia produced then reacts with water to form ammonium hydroxide, which creates a more alkaline medium and causes an increase in the pH of the urease medium. So as this happens, the phenol red, which is the pH indicator in the medium, changes color from yellow-orange to a bright pink color, as shown here. So a positive test result is indicated or shown as this, where the medium changes color from yellow-orange to a bright pink color. Whilst a negative Urease test result, the medium remains the same. That is, the color of the medium is still yellow-orange. Composition of urease medium. A typical urease medium contains the following components. 1. Urea, that serves as a substrate for the enzyme urease to hydrolyze to produce ammonia and carbon dioxide. 2. Peptone, that provides nitrogen, amino acid, and other nutrients for bacterial growth. 3. Sodium chloride, that maintains osmotic balance and supplies essential ions for bacterial growth. 4. Potassium phosphate, that acts as a buffer to maintain pH stability in the medium. 5. Phenol red, that acts as a pH indicator and changes color in response to pH increase due to ammonia production. And 6. Agar, that acts as a solidifying agent that provides a solid surface for growth of bacteria. The Urease test procedure, or step-by-step procedures involved in the Urease test. The first is to label the urea agar tube with the sample identification, the date, or other necessary information as shown here. 2 is to flame to sterilize the inoculating loop and allowing it to cool. 3. Using aseptic techniques, pick a well-isolated colony from the fresh bacteria culture as shown here. 4. Inoculate the slant by streaking the surface of the urea agar slant heavily from the bottom towards the top as shown over here. And one thing we also have to note in this step is that we must avoid tapping the agar. 5. Incubate the inoculated medium at 35 to 37 degrees Celsius for 18 to 24 hours. And what we also have to note is that if result shows negative, we must continue incubation up to 48 hours. This is because there are some bacteria that are weak urease producers and may require up to 48 hours for a positive result. Key important note: 1. Weak urease producers may require up to 48 hours for a positive result. And 2. Heavy inoculation is recommended to avoid false negative results. And over incubation may occasionally produce ambiguous reactions. So a positive result is indicated by a color change of the medium from yellow-orange to pink, and no color change indicates a negative result. The clinical importance of the Urease test. 1. Identification of Proteus mirabilis and Proteus vulgaris. This is because these bacteria are rapid urease producers and give a strong positive urease reaction. So this test basically is used to distinguish Proteus species from other members of the Enterobacteriocide. Diagnosis of Helicobacter pylori infection or H. pylori. H. pylori produces large amount of urease, allowing it to survive in the acidic environment of the stomach. Differentiation of Enterobacteriocide. So some urease positive bacteria are Klebsiella pneumonia, Morganella morgani, and Providencia stouti. And urease negative bacteria are E. coli, Salmonella enterica, and Shigella sonnei. So thanks for watching my video, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comment or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.